Hello, welcome to our lecture on the cell cycle, mitosis, and how we regulate all of those actions in the cell. Make sure that while you're going through this unit, and particularly the lesson that we're talking about today, go through your big ideas and make sure that you can see that link between what we've learned so far and what we're talking about today. And use these learning objectives as a way to test yourself that you're really understanding the material. So we're going to start with the cell cycle first. So you began life as a single cell. How did you go from that one cell to the incredible multicellular person you are today? Well, the answer is lots of division. You started out as that little zygote, and you kept dividing and dividing and dividing until you were a little embryo. And this is an embryo at about 53 to 60 days, or I'm sorry, 56 to 60 days. And then eventually you just kept adding more cells and you would grow. All of this is because your cells go through a normal life cycle, just like any other living organism. So the life cycle of the cell begins when it first divides. So as soon as that cell is separated from the other daughter cell that's created from that parent cell, then it's starting its own life cycle, and it's going to end the minute that it divides. And there's two major phases of the cell cycle, interphase and the mitotic phase, or M phase. These are going to be regulated by checkpoints, and it's key to understand that the length of this cell cycle does vary based on the type of organism you are, or the type of cell you are. So if you're talking about skin cells and intestinal cells, they're going to have a very quick cell cycle. If you're talking about, let's say, like liver or pancreatic cells, those might have a much longer cell cycle. Interphase is going to take up the majority of the cell cycle. It's about 90% of the cell's life. And this is where the cell is being the cell. It's going to grow, it's going to perform its functions, we're going to have transcription and translation occurring. It's going to do its job as whatever cell it happens to be. So if it's a skin cell, it's going to be protecting you. If it's a neuron, it's going to be firing and causing those action potentials that make you think. And in the background, it's going to be preparing for M phase if it's the type of cell that's going to divide. We're going to learn that some cells don't. And interphase is broken down into three parts. G1, or gap 1 phase. This is where the cell is going to do the majority of its growth. It's going to copy its organelles, and it's going to prepare to do S phase. This is where we will synthesize new DNA. Essentially, we undergo DNA replication, the part that we learned about with helicase and polymerase and all that stuff. And then we're going to enter G2, gap 2 phase. And this is where the cell is going to continue to grow, but it's going to, in earnest, prepare for M phase, that mitotic phase, where it's going to be dividing. Now, in near the end of G1, a cell can leave the cell cycle entirely and enter this phase that we call G0, the resting phase. It's essentially used for a couple different reasons. One, there's certain cells that will never divide. So instead of staying in the cell cycle, they leave the cell cycle. And they will spend their whole life in this G0 phase. Things like your nervous cells, your brain tissue, your heart cells, your muscle cells, they don't really divide, if ever. So we want them to leave the cell cycle so that there's not a chance of them dividing when they're not supposed to. Now, there are some types of cells that can then re-enter the cell cycle, like your liver cells. When they get certain signals from their environment, they're going to be able to say, oh, okay, I'm done resting. I'm going to re-enter the cell cycle and proceed to mitosis. And the smallest part of the cell cycle is mitosis, the M phase. This is where cell division is going to occur. And it's only about 10% of the whole cell's life cycle, but it's broken down into a bunch of different stages: prophase, prometaphase, metaphase, anaphase, telophase, and then an additional step that is not technically part of mitosis itself called cytokinesis, where we actually split the cell into two different parts. Mitosis, as we're going to learn, is really more focused on the DNA and the nucleus division. So here is just a graphic for you to use to study that goes into a little more depth of every single part of this cell cycle. Now we're going to talk about mitosis. This is cell division, where we're actually splitting the nucleus of the cell and creating two new daughter cells. So mitosis is going to result in two identical daughter cells. This is a form of asexual reproduction where the cell is essentially cloning itself. And it's going to occur in our somatic cells, our body cells. So how do I make a new skin cell? I need to divide it. And I want two identical skin cells. I don't want my skin cells to start diverging from each other. I want them to all be my skin cells. So in order for this to happen, we're going to have to replicate our DNA. We're going to have to copy it during that S phase. This way I can have one copy that I can put into one new cell, and the second new cell that I'm creating can have the second copy. And just as a quick review, humans have 23 pairs of chromosomes, giving us a total of 46. One set is going to come from our mother, one set is going to come from the father. So you can see here, this is chromosome number 1. And I have one copy that I got from one parent and the other copy that I got from another parent. And these two chromosomes are going to have identical genes within the chromosome. However, the two copies that you have are going to have slightly different versions of those genes. So maybe the gene will have hair color, eye color, height, all encoded within that DNA on that chromosome. But your mother may have given you the copy of the hair color gene that says brown. Your father may have given you the copy of the hair color gene that says blonde. And then we're going to learn in other units how we actually determine what your phenotype or your physical appearance is going to be. So it's in mitosis that we take all of these copies of all 46 of our chromosomes and we split them up. So we end up with the same chromosomes in every single cell. Now the purpose of mitosis is going to vary by the type of organism you are. If you're a single-celled organism, this is how you reproduce. This is how you produce offspring. If you are a multicellular organism, you need mitosis for a lot of different reasons. You need it to grow. We don't grow by making our cells bigger because cells are limited in the size that they can become. If they become too big, they're going to have too large of a volume and they're not going to be able to they're not going to be able to quickly move nutrients in and out of the cell. They're not going to be able to evacuate waste quickly enough and they can eventually die. They most likely won't even be able to get enough nutrients to sustain that size of a cell. So in order for a multicellular organism to grow, it's going to have to add more cells. We also, as multicellular organisms, need mitosis in order to replace damaged cells. So if you get a cut in your skin, the way that cut heals is through mitosis. We just produce more and more skin cells to heal the skin cells that were damaged or removed. Also, your cells have a set lifespan. They don't live forever. So cells that become worn out and die naturally need to be replaced, and we have the tools in mitosis to get that done. Now, before we get into the complex details of mitosis itself, we need a little bit of a review of our DNA structures. So remember that DNA wraps around these proteins called histones, and when the proteins in the DNA are in a complex together, when they're bound to each other, we call that chromatin. Now chromatin can be relaxed, where it's not very tightly coiled, or it can tightly coil into these packages called chromosomes. We also can call them chromatids, as we're going to see on the next slide here. So when we're doing mitosis, all of our DNA has to be in this tightly coiled form. We want our DNA to be easily manipulated. We want to be able to move it around without causing breakages or mutations or loss of DNA, essentially. We don't want to damage that genome while we're moving our chromosomes around. So after we duplicate the genome, we have a copy of every single chromosome. We start out with a chromosome looking like this, and once we duplicate it, we have 2 copies of that chromosome, and they actually pair up. There's a narrowing point on every single chromatid. When they're in a single form like that, they're either a chromosome or a chromatid, and that narrowing region is called the centromere. And this is essentially just a sequence of DNA where we have a repeated sequence of bases. And it's where the 2 chromatids that have just been copied are going to most closely associate with each other, where they're going to bind closest. And when they're in this form, where one identical copy is attached to the other identical copy, we call them sister chromatids. And that's just more relating to their relationship. It's not implying gender or anything else like that on them. It's just saying they are identical. So when they're paired up, we call them sister chromatids. And they're also going to have these structures on them called kinetochores. And these are usually going to be facing in opposite directions of each other. So that way the cytoskeleton can attach to this point on the chromosome and manipulate the chromosome. Now, mitosis requires the use of the cytoskeleton. The cytoskeleton is going to be coming in, grabbing these chromosomes and moving them around, splitting them apart and taking them where they need to be. The entire structure of all these cytoskeletal protein fibers is called the mitotic spindle. And it's going to start at this region called the centrosome. It's essentially just an area inside the cell where all of these tubules are going to be organized and regulated and moved around. It's essentially where the main control hub of the mitotic spindle is. These microtubules that are going to be created in the mitotic spindle, they can grow and they can be broken down. We call it polymerize and depolymerize. And this is going to be key to them to move things around the cell. They actually shorten if they want to pull something closer to the centrosome. And they actually grow when they're trying to lengthen the cell or push something away from the centrosome. Now inside that centrosome, which is just an area in the cell, we see these 2 structures called centrioles. And in animal cells, they're always there. And we're not 100% sure what their purpose is. We know that they are always there, but we know that if we remove them, mitosis occurs normally. We also know that they're not in plant cells. We don't have these structures in a plant cell, and yet a plant cell divides normally just like any other cell. So we're not quite sure what the function of these are, but they're present every time we do mitosis. So here's just a more detailed picture that shows you where we have the centrosome with those centriole pairs inside and all of the spindle fibers coming off of that centrosome. And this is a real electron microscope image, and you can see the actual spindle fibers, the microtubules inside that cell. The darker colored ones are chromosomes, but the little hair-like structures, those are the actual spindle fibers attaching to the chromosomes, and they're going to be moving them around. And here we've actually created fluorescence with our spindle fibers and our chromosomes so we can see them light up underneath a microscope. And this figure here shows all the different steps of mitosis. And these ones on the side here actually show you what is what. So the green right here, those are the microtubules. The blue, those are our chromosomes. And the red is actually mitochondria. So now let's get into the nitty-gritty of mitosis. The very first phase is called prophase. This is where our chromatin is going to very tightly coil or condense to form those visible chromosomes. The nucleoli are going to disappear, and the mitotic spindles begin to form. So you can see here our centrosomes are pretty close together, and they're all on one side of the cell. So we have to get those centrosomes to migrate, to move towards opposite poles of the cell. We want them directly opposite each other, as far away as possible, so that way when they pull the chromosomes towards them, we get those chromosomes as separated as possible. Now, prometaphase is sometimes considered just part of prophase, but it can also be broken out just because there are very specific things that occur later in prophase, and it's probably best to say that it is its own phase. The nuclear envelope will break apart, and those kinetochores will start to form. The kinetochores are where the spindle fibers are going to attach to the chromatids. It's where they're going to actually move them around. So we've got our chromosomes moving now towards the center of the cell, and our microtubules are going to start to attach to those kinetochores. Our spindle fibers attach to the chromosomes and help them to move around in the cell. And our centrosomes are going to be getting to their final destination here. They're going to continue to migrate to opposite sides of the cell. Now, interestingly enough, spindle fibers that do not attach to a kinetochore, they're called non-kinetochore spindle fibers, are going to attach to each other. And their job is to grow and push back on each other and actually lengthen the cell, because we're trying to split this cell into two new cells. So the larger the cell is, in this case, the easier it'll be to separate it out. Now, metaphase is where all of our chromosomes line up on this imaginary line in the center of the cell. We call it the metaphase plate. And it's really important at this point that all of our spindle fibers are attached to kinetochores, because if a chromosome isn't connected to a spindle fiber, it's not going to be moved around properly, and then we're going to have a cell that either has too much DNA or not enough. We're going to have potentially broken copies of chromosomes, which we want to avoid. So we're going to have spindle fibers on opposite sides attached to the chromosomes. So that way when we break them, we can pull them to opposite ends of the cell. Now, we have a checkpoint in the cell at this point where we're actually going to make sure that all of these kinetochores are attached properly and that our DNA is ready to be split apart. We really want to make sure that everything is good to go before we move into the next phase here. So anaphase is the shortest phase of mitosis. Our chromosomes are actually going to break apart. The sister chromatids separate. And there's these proteins called cohesin that hold them together right around near the kinetochore. Those cohesin proteins are going to be broken down, and the spindle fibers are then going to pull the individual sister chromatids apart. So in anaphase, all of the genetic material is going to be separated and pulled to opposite poles of the cell. So, like I said, our kinetochores are going to bind those microtubules. And it's at the kinetochore that we actually break down those spindle fibers. We make them shorter at the kinetochore, not at the centrosome side. So there's actually structures inside the kinetochores that will break off little pieces of that microtubule in order to shorten it. And when we look at our chromosomes and what's happening during mitosis here, I have one sister chromatid and a second sister chromatid connected to each other. When they break apart, I end up with 2 identical chromosomes that are now separated. This structure here is considered to be one chromosome. When they are separated, they are now 2 chromosomes because they contain identical information. The very last phase of mitosis is telophase. This is where the chromosomes fully reach the opposite poles of the cell, and we're going to essentially do the opposite of metaphase, or I'm sorry, of prophase and prometaphase. We're going to remake the nuclei around those separated chromosomes. We're going to make the spindle fibers disappear. The centrosomes are going to break down and no longer be needed. We're going to have our chromosomes loosen up. They're not going to be as tightly coiled anymore. And we're going to have essentially 2 nuclei in one cell. In order to now create 2 new cells, we have to go through the step of cytokinesis. Cytokinesis begins during telophase itself, so they're essentially happening simultaneously. And cytokinesis is the division of the cytoplasm and the membrane itself. It's going to be a little bit different in animal versus plant cells because of the presence of a cell wall. So let's go into that right now. Animal cells will undergo something called cleavage. We take these actin proteins and we create a ring around the inside of the cell membrane, around the entire inside of the cell membrane, in between the 2 new nuclei that we've created. And we're essentially going to pull that like a drawstring. The actin is going to get tighter and tighter, and it's going to pinch the cell membrane in on itself. And that forms what we call a cleavage furrow. And as it's going to keep getting tighter and tighter until the cell is actually going to pinch in 2 and create 2 new cells as a result. Now, we obviously can't do that in plant cells because they have the very rigid outer cell wall. So we have to build a new cell wall from the inside out. Plants will form what's called a cell plate. You can kind of see the little shadow of the cell plate here. The Golgi apparatus creates vesicles that move towards the center of the cell in between the 2 new nuclei. And inside these vesicles is a material that's full of cellulose and other building blocks of the cell wall itself. And they're going to actually create a cell wall inside that vesicle. As more and more vesicles come together, they're going to fuse together and create one big long vesicle containing an early cell wall. And finally, we're going to have so much of those vesicles come together that it creates a separation between the 2 cells. They're actually going to form their own cell membrane and cell wall as a result. So I have a few pictures for you guys. Take your time on the PowerPoint PDF that I'm going to attach and go through these and make sure you can recognize every one of the stages. This one and the previous one were animal cells. Here is a plant cell where you can actually see the cell plate right here. And this is a sample of an onion root, and you can actually see all of the different phases of the cell cycle at once occurring inside this sample of cells. Now, binary fission is the term that we give to prokaryotes undergoing division. They don't undergo mitosis in the same way that a eukaryotic cell would. So our archaea and our bacteria are going to asexually reproduce through this binary fission. The cell is first going to double in size, and then the DNA begins to replicate. And remember, prokaryotes have that circular chromosome, and at one point, it's called the origin of replication. That's where DNA replication is going to begin, and it's going to happen in opposite directions until the entire circular chromosome is duplicated. Well, as this is happening inside the cell, the origins are going to move in opposite directions. As we make a new chromosome, they begin to separate. One origin is going to move this way, and the other origin is going to move the opposite way. And that's going to naturally separate the chromosomes. And They do this all without spindle fibers, but the bacteria have their own version of the actin proteins that are going to move all this stuff around. So it's not quite the same as ours, but it's a really good early precursor of it. And they will pinch in their cell membrane, very similar to cleavage in animal cells, and then create 2 new daughter cells. Now, how does the cell regulate all of this? What happens if there's a mistake? What happens if they need to speed up the cell cycle because they're wounded and we need to induce healing? We're going to get into all of that now. So organisms have to have very precise control over every single point of this cell cycle. We grow very quickly when we're in utero. We grow very quickly as infants. Then we have a growth spurt in adolescence, and then we tend to maintain the same size for the rest of our lives. How does the body and the cells in the body know when to speed up and when to slow down that division? Then you can look even further inside the bodies and say, well, I have many different types of cells. My skin cells and my intestinal cells are continually dividing. And not only that, they can speed up or slow down division as needed. So they can speed up division when I get a cut from, a paper cut or something like that. They can speed up division if I have a damage to my intestinal walls. And then I have, within the same body, some cells that are never dividing. How do I exhibit such precise control over my cells? It all comes down to these checkpoints. We have a cell cycle control system, and there's 3 main checkpoints. There's a lot of others that we're discovering. However, these are the ones that tend to be the most important, and they're going to be the ones that we know the most about as well. There's one checkpoint near the end of G1. So we're saying this is the start of the cell cycle over here. So we have a checkpoint at the end of G1, we have a checkpoint at the end of G2, and we have one inside M phase, usually during around the metaphase stage. We want to make sure everything is good to go before we separate those sister chromatids. So the G1 checkpoint, this is probably the most important checkpoint because it's going to be the one that as soon as we give the green light here, the cell usually divides. If we don't give the green light, the cell won't divide. The other checkpoints, it's not as dramatic of a decision. When we look at cells that are given the go-ahead at G1, nearly all of them are going to end up dividing successfully after that point. So this checkpoint is looking to see if the cell is ready to replicate its DNA and enter S phase. If I'm ready to replicate my DNA and expend all that energy, I'm usually ready to separate that DNA as well. However, if we do not pass this checkpoint, there's going to be a rest phase that occurs here, that G0 rest phase. So the cell will be able to leave the checkpoint or leave the cell cycle, and either meet the criteria to divide and re-enter the cell cycle, or it's going to remain in G0 if it's the type of cell that does not divide. Now the G2 and the spindle checkpoints, or the M checkpoints, these are going to be to make sure that DNA replication occurs properly. And then in M phase, it's going to make sure that all of those spindle fibers are attached to the kinetochores properly. All of my DNA is lined up properly. I'm going to be able to separate those sister chromatids with no issues. Now, how does the cell know what to do and when? Well, in 1970, an experiment at University of Colorado found that there has to be some kind of chemical signals inside the cytoplasm, and as we found out later, some from outside of the cell as well, that can activate or stop the cell cycle. What they did was they took 2 cells in completely different phases of the cell cycle. One is in S phase and the other is in G1. So this one in S phase has been given the green light in that G1 checkpoint to go ahead and start dividing. This one may not have reached that point yet because it's still in G1. When we caused those two cells to fuse together, we found that immediately this second cell entered S phase. And the same thing when we did it with a cell in M phase, in mitosis, immediately those cells did what they needed to do to replicate their DNA and get into mitosis as fast as possible. So there was some signal inside this first cell that was telling the other cell, hey, it's time to divide. Well, these signals, these activating signals, are things like cyclins and CDKs that we're going to talk about. There's internal factors called promoting factors, and there's external signals that we call growth factors. And all of these are going to operate under the same mechanism that we learned with the cell communication unit. We're going to have something trigger and bind to a receptor and start one of those phosphorylation cascades. We're going to have one enzyme trigger the next enzyme, trigger the next enzyme to cause a change in the molecule. And eventually we can cause the DNA to be expressed. We can activate gene expression, or we can maybe even turn off gene expression depending on whether we want the cell to divide or not. So these internal signals we discovered are called cyclins. And these are just regulatory proteins, so they have to be expressed by the DNA. They have to go through transcription and translation. And when we make them, they stay inside the cytoplasm, and they will bind to these enzymes called cyclin-dependent kinases. We call those CDKs. When they bind, they're going to activate the kinases. And they're going to create that phosphorylation cascade. So that cyclin CDK complex, when they bind together, they're going to have the ability to now go and phosphorylate another chemical and create that, trigger that cascade of phosphorylation. Now we also have special kinds of cyclins. We have M cyclins that are processed in that M phase checkpoint, S cyclins during synthesis phase, and we have G1 cyclins. So we have a lot of different versions of these proteins to have control over very specific points within the cell cycle itself. And this is just a review to kind of jog your memory about what I was talking about with that phosphorylation cascade. That first activated molecule will work with the protein kinase to get activated, and then that'll activate the next protein kinase, which can activate the next one, and it's going to end up with an activated protein that is now going to cause transcription and translation or gene expression to occur, or maybe it's going to activate a repressor protein that's going to bind to the operator and not be able to produce that protein anymore through transcription and translation. And these cyclins are called cyclins because they have this fluctuation of activity. We see a lot of them during mitosis, and then they drop in number during interphase. So we can see that they're directly responsible for creating this mitosis occurring in the cell. Now we also have external signals. Maybe something outside of the cell binds to a receptor and triggers this phosphorylation cascade. One such of protein is called a growth factor. And growth factor is released by cells to stimulate the cells around them to grow as well. You had that example in your textbook of platelet-derived growth factor, where when your body senses that there's been damage to the cells and platelets respond to the area, they release this growth factor that triggers all of your cells to begin mitosis to heal whatever damage occurred. These external signals are density dependent. So if I don't have a lot of cells in an area, like if I get cut and now my cells realize that there's not other cells around them, they're going to trigger mitosis by releasing this growth factor. However, when the cells recognize that they're being touched by cells on all sides of them, they're not going to be releasing as much growth factor. They're not going to have enough growth factor to trigger division in all of these cells. We also have an anchorage dependence. Cells don't like to divide unless they're anchored to something like another cell or connective tissue or something like that. These are ways to keep our growth in check. Now, cancer cells, we're going to talk about those a little bit more in a minute, but they're not going to heed these warnings. They don't need to necessarily stop growing, so they don't care if they're already touching another cell. They're just going to keep ignoring those signals and keep dividing. So now cells that will fail checkpoints can undergo apoptosis, where the cell tells itself to destroy itself. It's programmed cell death. Another unfortunate consequence of the failure of these checkpoints is cancer. So if I lose control over my checkpoints, I'm losing control over the cell cycle. If I can't control my cell cycle, my cells are going to divide out of control. That's what cancer is. It's an uncontrolled growth, an uncontrolled cell division. They don't go through the proper checkpoints. They won't copy their DNA properly. And they're going to be essentially stuck in divide, divide, divide mode. Now, benign tumors, these are ones that are at the original site of where that mutation or that checkpoint failure occurred. And we can go in usually with these benign tumors, cut them out, and we're done. The cancer's gone. Malignant tumors are those that started out where that checkpoint failure occurred, but now maybe the tumor has grown so large or it's invaded a blood vessel or a lymph vessel, and cells of that cancer tumor have broken off and they are now traveling around your body. When they decide to anchor somewhere and begin to form a new tumor in a different part of your body, that's called metastasis. So when your cancer is spreading to other parts of your body. We have actual genes that we've now linked to causing cancer within our body. Proto-oncogenes are early cancer genes. That's essentially what that word means. And these are genes that create growth factors. So you can understand why, if there's a mutation inside a proto-oncogene, I can end up with cancer. Because if I create too many growth factors, my cells are going to be dividing out of control. On the other hand, too, if I mutate them so I can't create growth factors, that's also damaging to the cell. But usually what happens with these proto-oncogenes, they get switched on. Now all of a sudden a cell is going to be producing too much growth factor, and all the cells are going to be kind of tricked into dividing. An example is the RAS gene, and this is going to activate cyclins. So we're going to be expressing cyclins, making a lot of these proteins, and they're going to flood the cell, and we're going to now have all these signals to enter mitosis, and the cell is going to be continually wanting to do mitosis. We also have tumor suppressor genes. So these are genes that are there to stop cell division in case something isn't going right. If they are mutated, they can end up being in the off position, where now we can't make this protein that's going to stop my cell division. An example of this is the p53 gene. This one is a really important gene because it controls that G1 checkpoint. If this gene is mutated, then we don't care about that G1 checkpoint anymore, and the cell is just going to be continually dividing. On top of it, we can have viruses that cause cancer. We call these like communicable cancers, where I can spread them around. Things like the Gardasil vaccine that we have to get now that protects against the cervical cancer. These viruses will take their genome and incorporate it into our genome, like those retroviruses that we talked about. They'll create that provirus inside our genome. Well, if they happen to accidentally insert their genome into the p53 gene or something like the RAS gene, now those ones are going to be mutated, they're not going to be working properly, and the result is going to be cancer. So We've only recently learned about these, and they're a huge area of study right now. And how do we treat cancer? We've talked about this a little bit, so I'm just going to go through this quickly. We have chemotherapy and radiation are the two main ways, and these are just methods that target actively dividing cells. Chemotherapy are chemicals that will specifically target DNA replication, mitosis, and cytokinesis. But if we're killing rapidly dividing cells, we're going to have our skin cells and our intestinal cells be kind of collateral damage. So we're going to have hair loss, you're going to see rashes, you're going to get that vomiting because you're damaging healthy cells that are just dividing rapidly as well. And that's all I have for you. Hope you guys enjoyed this. If you have any questions, just contact me and let me know.